Hi friends, good morning. Next video from Dhruva Edutainment and Vivo classes. This is 4 marks question friends, short question from senior or intermediate second year zoology. From Unit 8 friends, applied biology. Friends, this is general question. Okay. What is our question friends? Write in brief the types of gene therapy. Friends, this is latest technology friends, what we are using to control the diseases. Write in brief the types of gene therapy. You have to know all these things friends. Gene therapy. You know friends, gene is present on the chromosomes. It has DNA. Okay friends. What is therapy? Therapy means what friends? Treatment or cure or remedy. Please don't write this yellowish color one in your IP friends. Therapy means what friends? Treatment, cure or remedy. So what is gene therapy friends? It is a therapy to control or to cure the genetic disorders. It is used in what friends? To control or to cure the genetic disorders. Okay friends? Now come to our definition friends. What is gene therapy? Introduction of normal genes into cells. You know friends, genes are present in the cell, particularly in the nucleus. Here what we are doing? We are introducing normal genes into cells. Why we are introducing those normal genes friends? In place of defective or missing genes. Due to genetical disorders, that individual may have defective genes or some missing genes. In place of those defective genes or non-functional genes, or in place of those missing genes, we are introducing healthy genes or normal genes. For what friends? In order to rectify genetic disorders is called gene therapy. Friends, I think you are following me. Once again friends, introduction of normal genes into where friends? Into cells. In place of defective or missing genes. Defective or missing genes in order to rectify genetic disorders is called gene therapy. Friends, here we are introducing normal genes or healthy genes from outside into an individual who has defective genes or missed genes due to genetic disorders. To cure those genetic disorders, in place of defective genes or in place of missing genes, we are introducing normal genes or healthy genes into that individual. Once again friends, introduction of normal genes into cells in place of defective or missing genes in order to rectify genetic disorders is called gene therapy. Friends, ఒకసారి తెలుగులో విందాం ఫ్రెండ్స్. జీన్ థెరపీ అంటే ఏంటి ఫ్రెండ్స్? జన్యు చికిత్స. ఒక వ్యక్తి పుట్టుకతో డిఫెక్టివ్ జీన్స్ని కలిగి ఉన్న, కొన్ని జీన్స్ని కోల్పోయి పుట్టి ఉన్న, ఆ డిఫెక్టివ్ జీన్స్ స్థానంలో, ఆ మిస్సింగ్ జీన్స్ స్థానంలో మనం కొత్త జీన్స్ని, ఆరోగ్యకరమైన జీన్స్ని ఇంట్రడ్యూస్ చేసే పద్ధతిని ఏమంటాం ఫ్రెండ్స్? మనం జీన్ థెరపీ అంటాం. ఓకే ఫ్రెండ్స్? Or you have to write one more definition friends. It is a technique, friends, it is a technique that involves inserting normal genes into an individual cells to prevent a genetic disorder is called gene therapy. Friends, it is a technique in which what we are doing friends? We are inserting normal genes into an individual cell to prevent what friends? To prevent a genetic disorder is called gene therapy. This gene therapy is of two types friends. What is germline gene therapy? Somatic line gene therapy. This gene therapy is classified into two types: germline gene therapy, somatic line gene therapy. Friends, germline gene therapy, somatic line gene therapy. First, you know what is germline, what is somatic line. You know, friends, germplasm and somatic plasm. Germplasm indicates what, friends? Sex cells or reproductive cells. Somatic cells indicate what, friends? Other than sex cells. So germline gene therapy, somatic line gene therapy. Germline gene therapy means what, friends? Here we are introducing new genes into the germplasm, means that is either in the sperm or ova. Somatic line gene therapy means what, friends? We are introducing new genes other than sperms and ova, means in the normal body cells. So when we introduced new genes into the germplasm or in the through ova or sperms, it is heritable, friends. Heritable means these characters transfer from one generation to other generation. But somatic line gene therapy is non-heritable or inheritable. Okay, friends. Now see one by one. First we go to the germline gene therapy, friends. Germline gene therapy. Here you know what is germ means. Here germline, germplasm means what, friends? The cytoplasm or the plasm present in the ova or sperms and the reproductive cells. You have to remember it, friends. Okay. Germ means here not microorganism, friends. Okay. Now come to here, friends. Germline gene therapy. It involves inserting normal genes into an individual's sperm or ova. See, friends. Here where we are introducing the normal genes are introduced into an individual's sperm or ova, friends. You know, sperm or ova are gametes. They have germplasm. That will be passed, this germplasm, these genes will be passed down to future generations. See, friends, when we introduced healthy genes or normal genes into sperm or ova, those genes may inherit from one generation to other generation. That is only, friends, here. It involves inserting normal genes into an individual's sperm or ova that will be passed down to future generations. These inserted genes, what is in the future generation? These inserted genes become a part of their genome. The changes are heritable, friends. The changes are heritable. Here heritable means what, friends? Inheritance. Those characters inherited from one generation to other generation. So due to germline gene therapy, we have an advantage, friends. What is that? The inserted genes may inherit from one generation to other generation. Here, friends, I have given the definition of genome. What is genome, friends? The sum total of organisms DNA is called genome. The total DNA of an organism is called as what, friends? Genome. Please don't write this yellowish one in your IP, friends. But you know what is genome? A genome is an organism's complete set of genetic instructions, or an organism's complete set of DNA is called genome. The total DNA of an individual is called as what, friends? Genome. Now, come to somatic line gene therapy, friends. You know, friends, what is somatic? Other than gametes, means other than ova and sperms. Remaining body parts, remaining cells are called what, friends? Somatic cells. Okay. Now, somatic line gene therapy. It involves inserting normal genes into an individual's somatic cells of a patient. Here we are inserting normal genes or healthy genes in the somatic cells of a patient, not in the germ cells, not in the ova and sperms. Okay, friends, other than gametes. Now, since these changes are inheritable, they won't transfer from one generation to other generation. They are inheritable, friends. Okay. Now, somatic line gene therapy is of two types. See, friends, gene therapy is of two types. One is germline gene therapy, and another one is somatic line gene therapy. The somatic line gene therapy again divided into two types, friends. What are they, friends? Ex vivo gene therapy. Second one is in vivo gene therapy. Friends, somatic line gene therapy is of two types: ex vivo gene therapy, in vivo gene therapy. Friends, here ex means outside, in means inside. Here you have to know vivo. What is vivo? Friends, vivo means within the living. Within the living. See friends, what is ex vivo gene therapy? In this, the cells are collected from a patient, modified outside the body and inserted back. See friends, in ex vivo gene therapy, what they are doing, friends? They collect the cells from the patient and they take them outside and they modify it outside the body, and later they inserted back into that patient. See friends, in this means in ex vivo gene therapy, the cells are collected from a patient. What are those cells, friends? Defective cells or missed cells. Cells with defective genes or cells with missed genes. The cells are collected from a patient, modified outside the body and inserted once again into that patient, inserted back. That is ex vivo gene therapy. Now come to example, friends. SCID. What is SCID, friends? Severe combined immune deficiency syndrome. Friends, SCID. Severe combined immune deficiency syndrome. That is best example for ex vivo gene therapy. Example what, friends? SCID. What is SCID? Severe combined immune deficiency syndrome. Now come to in vivo gene therapy, friends. In vivo. In vivo means what, friends? Inside the body. In this, the genes or cells are modified inside the body. In this, the genes of cells are modified inside the body of a patient. See friends, in this, the genes are friends here. The genes or cells are modified inside the body of a patient only. Friends, here not they are taking the cells outside. They are treating the patient within the body, inside the body of that patient, friends. So in this, the genes or cells are modified inside the body of the patient only. That is called as what, friends? In vivo gene therapy. Example, friends, cystic fibrosis. Friends, cystic fibrosis. Okay. Now here you see, friends. See friends, this is defective gene. See friends, here this green one, we are introducing what, friends? A healthy gene. Now this is modified cell, restored cell. This is called as what, friends? Gene therapy. Okay friends, now come to in vivo and ex vivo. See friends, in vivo, here they are treating the patient when the cells are inside the body only. See friends, ex vivo, here they are collecting the cells, they are taking the cells from the patient and they are treating. After treatment, once again they are inserting the cells. This is ex vivo germ therapy. This is in vivo gene line therapy. This is ex vivo gene line therapy. Okay friends, that is about what, friends? Gene line therapy. Friends, how many types of gene line therapy is there? Two types: germline gene therapy, somatic line gene therapy. Somatic line gene therapy, how many types, friends? Two types. What is that? Ex vivo gene therapy, in vivo gene therapy. For ex vivo gene therapy, example SCID. From in vivo gene therapy, example cystic fibrosis. Okay friends, this is about what, friends? Gene therapy. Once again you see, friends, what is the definition? Introduction of normal genes into cells in place of defective or missing genes in order to rectify genetic disorders is called gene therapy. Okay friends, Thank you. Good day, friends.